Okay, great. Next, I'm excited to introduce Dr. Kevin Yang. He's a senior researcher at Microsoft for an in-person talk.
Thanks, everyone. So today I'll be talking about multimodal machine learning for protein engineering. Is that close enough to the mic? I hope. All right, so briefly, the protein engineering problem. So when we talk about proteins, these are sequences of amino acids, and there's 20 or so naturally occurring amino acids, and we can represent them using the letters of the alphabet. And they come out as a sequence, and that primary sequence or structure falls into this three-dimensional structure most of the time. And that structure, or this folding process, is still an object of study. And obviously with AlphaFold 2 now we can sometimes predict what structure a sequence will take. But even then we don't understand necessarily why that is or how it gets there. And that structure mediates function most of the time, unless we're talking about intrinsically disordered region. And how you get from structure to function is even more difficult. But for protein engineering, what we care about is what sequence will give you the desired function. So you start with the function you want, and you want to go backwards and invert that and come up with a sequence. This is a fairly hard problem. In fact, it's so hard that if you sidestep it and can figure out a way around it, they give you a Nobel Prize. So in directed evolution, what you do is you start with a parent protein that kind of does the thing you want. So on the right, I'm representing that by a starting point at blue dot on a fitness landscape that's kind of off to the side. So it's on the landscape, but it's not at one of the peaks. Then you randomly mutate this protein to come up with a pool of variants, and you screen those. And that's by taking random steps, random tiny steps in random directions. You pick the one that improves it, throw away the rest of the information, and repeat until you're happy with your results. So to do this, you need a starting point. And you also need a relatively high throughput screen, unless you want your PhD to take 60 years. And a very low bar for high throughput screen is about 100 a week. You would prefer to do thousands or tens of thousands a week if you can. So I mentioned earlier, when we make those variants, and then we measure them all, and then we take the best one as the new parent, that means we're throwing away all the information in every other variant you make. So if you take all that information instead, you just have to train a sequence function model. And instead of just taking the best one and randomly mutating it again, you use that model to somehow choose the sequences you want to measure next. You can do this much more efficiently. So a quick case study here, we're going to design some channel rhodopsins with me and Claire and Austin, whose picture is down there, who did all the experimental work, except for the one day I watched Claire patch clamp a bunch of things. I think I cloned some things too. So channel rhodopsins are these 7 transmembrane helix proteins, and they bind this molecule called all-trans-retinal. And it's in the ribbon structure. You can see it. It's that gold molecule inside it. That gray represents the membrane. You can count the helices if you want. And when usually the channel rhodopsin is closed, when the right wavelength of light hits it and activates retinol, it shifts conformation and causes the channel to open. And this allows usually sodium ions to flow into the cell. And the native function here is to move towards or away from a light source. And these were originally discovered in algae. However, it turns out if you can express these in neurons, you can do something called optogenetics. Opto for light and genetic because it's genetically encoded. And this, once you've convinced your neurons to express these proteins in the membrane, if you shine the right wavelength of light on those neurons, you'll activate or deactivate those neuron populations depending on which channel rhodopsin you put in. And that means you can do things like make your mice forget that they're scared of some corner of the cage or they get shocked, or make them turn left or right when they're running. Basically mind control. For obvious reasons, we do this in mice and not in people. So if I'm designing a channel-rhodopsin or engineering a channel-rhodopsin for optogenetics, I might have some requirements. For example, I really needed to localize in the mammalian cell membrane. Remember, these come from algae. They're not always expressed well in mammalian cells. If they are, they may not localize properly to the plasma membrane. And if they don't, that means they don't, obviously, if it's not in the membrane, opening or closing doesn't matter. It's not going to let anything into the cell. And here we have an assay where only channel rhodopsins that are correctly localized in the membrane so that the C terminus is outside get dyed green. Generally, for optogenetics, you want your channel rhodopsin to be as sensitive as possible. You want to be able to activate it with as little light as possible. And that's just because if you can use less light, you can do things like only shave the skull instead of taking the bone out. And if you're doing something like behavior modification, that takes away a confounder and it's just less work. You may want to create different activation wavelengths, and that would allow you to have different to have different populations of neurons in the same space that express different channel rhodopsins and get controlled by different wavelengths of light. You may also want different on-off kinetics. So you can imagine something that turns on or off really quickly could be useful, or something that turns on and pretty much never turns off. And we're going to optimize this using machine learning. So we start with not one or two, but 3 parents. And here I'm showing max photocurrent, which is we flash them with different wavelengths of light and see what the photocurrent is at the most activating wavelength. And the 3 parents are all around 1 nanoamp. They were chosen because one of them had a structure, one of them had great currents, and one was blue shifted and one was red shifted on their activation wavelength. We then designed a recombination library. And first we made all the single block swaps. This is not actually all the ones we made. These are only the ones that localize in the membrane. And even the ones that localize, most of these very non-aggressive variants don't have any photocurrents. But some of them are better than the parents, great. We also make some much more diverse variants. Most of these, we made, I think, 120 of these. There are not 120 points there because most of them don't localize. And the ones that do, almost all of them have no photocurrent. But there is one that's really good. It's almost 3 nanoamps. So there's no machine learning yet here. There's some stats to pick on the block swap to be as diverse as possible. And then we train a model on those first 3 columns, use that model to pick another group of variants. Here we got some improved variants, but there's also a lot of things that don't localize and don't have photocurrents. But we're learning. And finally, put all it together, we actually built a localization model and a current model and picked things that high probability of both. We also chose things that were redshifted or blue shifted or had fast or slow off kinetics. And we also chose those 2 purple points that are negative controls. So those are very close to one of those sequences that's really high from the first from the iteration 1, but are predicted by the model to have no photocurrents. And that's just to show that this is not a trivially easy problem. And the cool thing now is these are sensitive enough that we can control mouse behavior without removing the skull. And I'll show a quick video. And the fiber optic cable is glued to the skull, and we've shaved this. We've gotten rid of the fur and some of the skin, but we've not removed the skull, which is the previous standard. All right, so we've talked about this I call machine learning guided directed evolution, or MLDE, and some successes and limitations. So in that case, we trained accurate models with fewer than 200 measurements, which is great because these measurements require a patch clamp, and 200 measurements with replicates is a lot for patch clamp. And from those 200 measurements, we're able to find rare improved sequences. However, we still had to find a starting point. In fact, we had 3 of them. And the only data we ever used was data we collected ourselves, and that ignores all the data out there in protein world and everywhere else. So like I said, we have access to large protein databases now. For example, UniProt has 250 million sequences or so. There's actually a bigger one now called BFD. I'll let you figure out what the F stands for. And if you deduplicate UniProt so that to 100% identity, there's 236 million sequences. You can cluster those to 90% or 50% identity and get 116 or 42 million sequences. I think these numbers have gone up a bit since I made the slide even. If you're really picking, you only want things that have been experimentally verified, like SwissProt with 250,000 sequences. And some of these we have crystal structures for, about 169,000 in PDB. Of course, now with AlphaFold 2, you can predict structures for anything you want, and they're slowly doing this for everything. In your F90, I think they've already done most of SwissProt, if not all of it. Compare that with the 200, the measly 200 measurements we had for channel-rhodopsin. But keep in mind, those 200 were very high-quality measurements, and they were exactly what we want. But still, it seems like we should be able to get something out of all this. So now I'm going to come to kind of my central claim here, which is that we should use multiple data modalities in machine learning to design proteins. So the idea here is during your initial model training, you're going to look at natural protein data. That could include sequence. It might include annotations. There could be Go annotations, could be EC numbers, could be PFAM. You might also want to look at structure, either real, either experimental or predicted. Or you could look at substrates or ligands. You could look at free text from the papers that describe the proteins. And once you have that model, there's kind of 2 classes of things you can do with them. One is you can fine tune that model to predict whatever fitness function you'd like. Or you can use machine learning to generate de novo proteins that do the thing you want. So in the first one, you're putting in new sequences in the model, and your model spits out usually a number for prediction, how good it is at something you want to do. For certain de novo things, ideally you put in the desired function, and your model will give you candidates that do that function. So let's walk through a simple case of pre-training with just sequence to see how this might work. So early on, by early on, I mean in like 2016, a lot of methods we just borrowed from natural language processing. For example, me and some other people have papers where basically use word2vec. The hypothesis there is that similar chimers for chunks of amino acids occur in a similar context, similar meaning similar in chemical property or functional property. And you can use that by looking at surrounding chimers, 1, 2, 3 on each side, and use that to predict the center one. And you can use the intermediate there as an embedding space if you'd like, for representation. Then people came along and said, well, we can also factorize these sequences and go through all of UniRef or something and just given a sequence, predict the next amino acid given the previous ones. We call that an autoregressive language model. You can do that with a neural net. And you can do this, use this for generation or for pre-training. So you can think, if I've trained this on 15 million sequences, maybe if I just let it spit out random sequences, it will give me functional proteins. And there's some recent work that came out just last week showing that it does seem to be able to do that. They stole the architecture from GPT-2, which is a language model for English. And once again, you can use that penultimate layer to represent the whole protein or to represent each amino acid in the protein. And then there's an idea that we also stole from natural language processing called a neural mask language model. Now, instead of predicting the next token or the next amino acid, each time through, we randomly corrupt the sequence by either masking some positions or corrupting some positions, and then pass that through a neural net that looks in both directions instead of only looking in one direction. And we try to recover the original sequence. You can also use that penultimate layer as a representation. And usually the autoregressive language models and neuromass language models are done with what are called transformer models, which were proposed by a paper out of Google in 2017. It's an attention model, meaning that at every step, it learns what part of itself it should pay attention to come up with a better representation. You stack a bunch of those. Originally, it was proposed for machine translation, but now it's also used for these autoregressive language models and for these math language style things. And recently, people have also started using them for images and all sorts of other things. However, there is one disadvantage to these models, and that is that they scale really poorly with sequence length, because that attention goes through what's called attention matrix, which is a length by length matrix. So the computation and the memory requirements scale quadratically with length. We could also, and I was curious one day and I thought, well, we could do, there's a whole zoo of approximate transformer models out there where they try to get a linear dependence on length. But ConvNets, they're kind of out of fashion now, at least for language and sequences, but they scale linearly with length. And it turns out that if you go and find a fairly simple convolutional architecture, which is just embedded your sequence in the embedding space, then put it through a bunch of blocks that are just dilated convolutions, so you get a really wide perceptive field. And I'm going to call this CARP for convolutional autoencoding representations of proteins. And at a variety of model sizes, so on the X, I've shown log number of parameters, and they range from 4,000 to 640 million. But we're competitive with the state-of-the-art transformer mass language model. And this is under the pre-training task. So how well we recover those mass tokens. ESM1B is the state-of-the-art mass language model out of, it's a transformer, it's trained by Facebook. I didn't connect it to mine because they use a different training set than I do, a different evaluation set. But across the full number of parameters I've shown, we get comparable performance. And as expected, the CNN scales better with length. And here I'm showing loss by length. And ESM1B and its test set seems to do a little bit better, but not that much better. And they also max out. They don't accept sequences longer than 1024. I think I trained on sequences up to 2048, but you can put in as long as sequences you want, as long as you have to compute. So I evaluated it up to 4096. But what we really care about is, can I use this from protein engineering? And in other fields, like image processing or natural language processing, machine learning progress has really been driven by benchmarks. And that's true actually in protein world too for structure prediction with CASP. And what I care about is predicting fitness from sequence. And what I mean by fitness is the ability of a protein to do something that an engineer cares about. And it's going to be a quantitative measure usually. There's some previous work here called TAPE. And two of their data sets are engineering data sets, and they're fluorescence and stability, or they're called. One's a GFP mutation data set, one's a bunch of designed small proteins, and how you want to see how stable they are. So ESM, that Facebook transformer model, is a mass language model, and these are Spearman correlations. Seems to do okay. The original TAPE transformer, which is 20 times smaller, does about the same. This is a very small contrastive model, also does about the same. In fact, you can just do linear regression. And in one of those data sets, it does about the same. You can use a tiny CNN, this is like a 3-layer CNN on 1 hot encoding, also does about the same. And our new model also does about the same. So this is not really a great benchmark if you care about these sort of engineering data sets. So I worked with a bunch of people. Some of them are at Microsoft, some of them are at Caltech, some of them are at Berkeley, some of them are at MIT, Salesforce, Technical University of Munich. And what we did was we chose some new landscapes that other people have measured. We chose interesting ways to do train test splits that measure generalization in ways we care about for protein engineering. And then we trained and predicted a bunch of baseline models that you can compare to. So the three landscapes are GB1. So this is a 4-site site saturation library. They captured almost all 160,000 possible variants. There's an AAV data set where they mutated 39 positions, including insertions and deletions. This is data out of Dino Therapeutics. And then we also found a thermostability data set where they measured thermostability for proteins across tree of life. So as we go left to right, we go broader in sequence diversity and less specific. We're going to split this in interesting ways. For example, the most challenging split you can do with GB1 is one versus the rest. You get you train a single mutant, you test and everything else. Less challenging is train a single and double mutants and test and everything else. Or you can do single, double, triple, test and everything else. You can do a random split, which is the easiest one. Or you can do train on the bottom 80%, test on the top 20%. And you can think for all of these, for example, if you only train on one, test on the rest, is something that comes up a lot if you're doing machine learning in a directed evolution lab, because they may have done a site saturation library, and you have that data, but they don't want to go, then they come talk to the machine learning person and like, can you help us before we go collect the full site saturation combinatorial library? And the last one makes a lot of sense because for engineering, you care about how well our model does at the really good things and at telling those things apart. Similarly for AAV, we do random design. So they had some sequences that were generated randomly and some that they had used simple models to design. Also can reverse that. And we have the random split. Then we have the single mutants versus the rest, double mutants versus the rest, 7 or 3 mutations versus 8 or more. And then the same low to high. For thermostability, we cluster everything. And then we do a split so that no cluster is split between train and test, so that you don't get really trivial generalization. On the test set, we only evaluate on one thing from each cluster to avoid biasing towards large clusters, but we keep all the sequences in each cluster during training on the train set so that we don't lose too much data. And there's also splits where you only get sequences from certain cell lines and predict on other cell lines. We evaluated a bunch of baselines, including Levenshtein distance and Blossom 62. These are very simple, not even machine learning models. Some supervised models with no pre-training, like ridge regression, or that tiny CNN, or ESM without training. And then also pre-trained ESM transformer models, pre-trained either UniRef 50 or UniRef 90. And the pre-trained models, ESM at least, does the best when you have very few data. For example, you only get a single mutant, so you have to predict everything else. Or on thermostability, where there's data from across tree of life and not very much of it. The supervised models are very competitive in everything else, especially the random splits where you have 30,000 training points or something. If you would like to try our models in these data sets, they're available as tidy CSVs. There's also a GitHub. I think the website tells you to go to the GitHub to download the data sets. And so that was all kind of a side quest to figure out how to evaluate my model. And I'm showing results here for GB1 on all the splits. And so there's a bunch of bars because you can just think of different ways to fine tune. You can fine tune the whole model, or you can fine tune only the last layer and freeze everything else, just much more efficient, but doesn't seem to work quite as well. So that's blue and green there. Naive means that there's no pre-training at all. So we're just evaluating the architecture. And then purple is ESM1B with the transformer frozen, only the regressor on top is trained, and then brown is the ridge regression baseline. So for low versus high, ESM does really well. But you can, for 1 and 2 versus the rest, you can see that CARP does slightly better than everything else. Actually, for all the other ones, you can see CARP is slightly better than everything else. Sometimes without pre-training. But I promised we'd be using multiple data modalities, and so far I've only talked about sequence. So let's add some more things. Here, let's add structure. I'm going to combine sequence pre-training with some structure information. So I think back in 2017 or 2018, John Ingraham published this paper where he actually did the autoregressive leading, but you can do this with a mass language model, where instead of just predicting the mask locations from everything else, you can predict the mask locations from the unmasked locations and from a bunch of structure information. So here I've drawn a simple structure. When you want to predict a location, you get sequence and structure information from its structural neighbors. You can do this with a graph neural network. So you start with 19,000 structures and sequences, corrupt the sequence, get known edge embeddings, pass it through a GNN, and recover the sequence. And in the pre-training task, this will give you a perplexity about 4.96, with only 19,000 training examples. To give an idea of how good that is, CARP with 640 million parameters and 42 million training sequences on the same test set will give you a perplexity of 7.06. But we can do even better if instead of learning fresh node embeddings, we just feed in CARP's node embeddings to the GNN. Now we get a perplexity of 4.08. So this is new ongoing work. I'm still working on fine-tuning on all those fit data sets. But I believe in talking about the latest and greatest. And then one other thing. So far we've talked about the pre-training paradigm with sequence and with structure. So now we're going to talk about the generation paradigm with sequence and annotations. And what we're going to do is we're going to generate single peptides. So single peptides are secretion signals. So proteins are made in cytoplasm. Sometimes they have a single peptide on them. They get exported through a sec channel, and then they get cleaved. And I don't know why I flashed briefly, but this was done at Caltech with a bunch of people, including Zachary Wu and Alicia Lee. And these secretion signals kind of have a canonical, I say structure, but that's not structure in the 3D sense, it's structure in the format sense, I guess. So on the M terminus, they're about 15 to 30 amino acids. They are at the N terminus. There's typically an N-terminal positively charged region, hydrophobic core that often is a helix, a polar C region, and then a terminal motif and a cleavage site. They control extracellular secretion in prokaryotes and secretion from the endoplasmic reticulum in eukaryotes. And they're dependent on both species and protein, and the rules I just given you are not sufficient to generate novel functional signal peptides. So we're actually going to use this transformer here and go back to the original transformer use, which is machine translation. And what I did here was I went through SwissPro, pulled out 40,000 protein sequences that were labeled with this, or annotated with a single peptide. And I'm going to pass the mature protein, the protein without the single peptide, to the encoder, and then pass information to the decoder that spits out the single peptide. And after training, the first computational measure was, can we trick a single peptide annotation model, which looks at the whole protein and gives you the probability that there is a single peptide and where the cleavage site is. So we start with some simple models. A hidden Markov model is one that just generates by looking at the previous state. And these are pretty bad. So higher here is better. The heuristic I just described earlier, just generating using that, is much better actually than the hidden Markov model. We also train what's called a variational autoencoder, which is another style of generative model. This one does even better. In fact, on signal P, it does better than the transformer that I'm going to focus on for the rest. But if we look at the VAE, most of the sequences it generates are very repetitive. And it looks like it's just really learned that things should start with M. And then in that hydrophobic region, there should be lots of L's, loose ends. So we actually made a bunch of these in a collaboration and put them into cells in the Bacillus subtilis in a few different classes of enzymes. And we also did the natural single peptides as a baseline. So 79% of those work. And 48% of the generated ones work, which is pretty good. And you always want that positive control baseline because this is molecular biology and nothing is ever perfect. And for the ones that work, the enzymatic activity level is comparable. All right, so now I'm going to talk a little bit about some future work. So first one is, when does pre-training actually help? So here I'm showing a plot of the task is predicting secondary structure at every location in the protein. This is the 3-class secondary structure. The colors represent how big the model is. So we have a 640 million parameter model, the 66 million parameter model, and a 600K parameter model. And the method is whether we fine tune the whole model or freeze the model and fine tune the top. And you can see generally the bigger the model is, the better. This line here is without any pre-training. And we find that as we pre-train the model more, so the pre-train loss gets lower, the downstream lot accuracy gets higher. This is exactly the behavior you want to see. Right? The better I do in the pre-training task, the better I do in the thing I care about. Unfortunately, as a protein engineer, I don't really care about secondary structure. I care about some downstream function, but this is great. Unfortunately, for most of the functional tasks, it looks more like this. I don't know what this is. So sometimes pre-training helps a lot. Sometimes it seems like you get a pretty good amount of lift just by fine-tuning the naive model actually. So who knows? And it's definitely not a nice smooth line. So this is a whole field of investigation. Maybe we need structure. Maybe it'd be nice to have a way to predict, given the task, whether your pre-training regime is any good at all. Some other future work that I'm working on. One is learning to generate enzymes that catalyze desirable new nature reactions. So we've heard a lot from Abby and from Sarah about how synthetic organic chemists have this almost magical ability to look at the reaction an enzyme does and think, this reaction is kind of similar. Nothing in nature does it. But maybe if you try some enzymes, one of them will be a good starting point for evolution. Unfortunately, I'm not a very good synthetic organic chemist, so I can't do that. So I'm stuck trying to train models to do it. And another idea is trying to generate smaller versions of proteins for easier packaging and downstream engineering. The canonical case here is CRISPR-Cas9, which is 1,300-ish amino acids. If you want to deliver it with, for example, AAV, it's a little bit too big, but there's lots of other examples here. With that, I've acknowledged some collaborators throughout. Someone acknowledged the biomedical ML group at MSR New England. Unfortunately, I joined during the pandemic, and so did everybody else, so we don't have a picture together. And now I'll take questions.
Okay, questions for Dr. Yang?
Yeah, so I have a question kind of about when you're looking at the different methods for producing train test splits that you showed, and I guess I'm not completely naive when it comes to like selecting these, but also I don't always know what the best way to do it is for a project. And so, for instance, when you do your low high split, you do the 80% at the low and 20% at the high. Is that just picked kind of arbitrarily by you, or is there some sort of way that you can set that limit to ensure that you get a good, robust model out?
Yeah, so the intuition comes in thinking that predicting high things from low might be a good thing to do. The 80-20 was pretty arbitrary. And I think we just did it to make sure we had enough sequences on both sides.
Okay, gotcha. So there's no magic.
Yeah, there's no magic there. And maybe for your task, you want something different, and you can go look at the raw data and do your own split.
Okay, thanks.
Really interesting. I'm wondering how successful or not was the application of natural language processing to finding little chunks of amino acid sequences that were redundant, and particularly were they able to be redundantly related to function? Where did all the dust fall on that project?
So I wasn't so much trying to find things that were redundant there, as I was just trying to come up with a good way to represent proteins as vectors, sequences of numbers, as vectors of numbers. So back when I did this, I basically did that project while I was waiting for the tower adoptions to be measured, and I had some data sets laying around. But it turns out that, I'm trying to think. Yeah, I've never really looked at how any chunk might be redundant in that way. I'm kind of thinking about it in this last project, but there, what I'm actually thinking more of is, given we train structure prediction models using an alignment, right? And some of those alignments will have deletions in them. So given the structure and where we know deletions have recurrent nature, can we predict deletions we can make to a given sequence that won't affect function? So that's how I'm approaching that more than a K-mer perspective. That makes sense.
Thanks.
Sorry, I wasn't sure which project you were asking about.
I just have a quick question. On the machine learning model used for the channel redoxing engineering, did you try that model for other proteins? See that whether it works with like a very small number of data points?
I haven't published anything on it. So we use a Gaussian process model there with a matern kernel. It turns out I just out of curiosity tried some other things. A random forest does about as well, but you don't get nice uncertainties out of it. I have, well, every so often I'll run a Gaussian process something else. Unlike those flip things, a GP with even just a square exponential kernel on the one hot encoding is fairly competitive with everything else, as you would expect. It's not necessarily better than anything else. I also Francis once asked me to review a paper when I was a grad student, and I was bored, and I thought their baselines were no good, so I trained a GP on it and also did better than their baseline. But when it just comes to you have a small data set or doing directed evolution, it's such low-hanging fruit that it doesn't really matter which method you use. It's better than throwing everything away. Like a lot of the earlier work from like 2002 and 2003 by, I think his name is actually Michael Fox. His last name is definitely Fox. Just use a linear model. And even a linear model is better than throwing everything away.
Okay. Yeah, you can go ahead.
Thanks for the great talk. Just a little bit add on that. What's your thoughts on like a generic model that fits all the different train test split that you mentioned, and also on different proteins, on different tasks, different fitness that you're interested in, or like every time you do a protein engineering task, like you face a new protein, new characteristic, and you develop a new model based on it. What are your thoughts on it? These two. a generic model?
So far, no one's really been able to come up with a generic model. If you go look at the paper on Flip, that benchmark data set, if you look at every split, we benchmarked ESM 1B and 1B and some small models and some baselines, and whether you freeze the pre-trained part or you fine tune through it. And basically, it's kind of random which one does best in any split, especially between the 4 pre-trained variants and even like naive transformer variants. So right now, with the architectures we have, there definitely isn't like a universally best model. There's kind of a new line of work that's kind of underexplored, kind of a meta learning line, where every time the idea would be you have a bunch of these tiny engineering data sets, right? And instead of training them separately, you get one, you train it, the model improves. You get a new one, you train it. It doesn't lose performance in the old ones, but it uses that everything that what it knows already to boost performance in your new data set. That would be the dream. I don't think we've gotten there yet.
Okay, join me in thanking Dr. Yang for his talk. Okay, and then happy to introduce Ocean Sui,